Welcome everyone. I'm Subhalakshmi, first MSc Microbiology, Sacred Art College, Autonomous Tirupattur. What we are going to see in this session is classification of viruses. Introduction to viruses. Definition. Viruses are microscopic infectious agent that require a living host cell to replicate. Characteristic. It is acellular in nature, composed of nucleic acid, either DNA or RNA, surrounded by a protein coated is called capsid. It can be either enveloped or non-enveloped in nature. Virus classification. Viruses are classified based on various factors: host, disease in human, target issues, mode of transmission, vector, structure, and nucleic acid. Viral genomes, nucleic acid, DNA or RNA, standardness, single stranded or double stranded, sense and method of replication determine its class. Other classification are determined by the disease caused by a virus or its morphology. Viruses can be replaced in one of the seven groups. Importance of virus classification. Purpose of classification. Understand viral structure and function. Aid in diagnosis and treatment of viral infection. Facilitate research and vaccine development. Criteria for classification. Key factors. Type of nucleic acid, DNA and RNA. Structure, enveloped or non-enveloped. Shape, icosahedral, helical, complex. Host range, plant, animals and bacteria. Classification based on the structure of virus. Structurally, viruses are of three types: helical symmetry, icosahedral symmetry, complex symmetry. Helical symmetry. The protein and nucleic acid are arranged in a spiral. Example: tobacco mosaic virus, TMV, rabies influenza. Its TMV is look like a bunch of banana arranged. Most helical viruses are enveloped and RNA viruses. Icosahedral symmetry. The protein subunits from a symmetrical shell around the nucleic acid core. An icosahedral has 12 vertices, 20 faces, and 30 edges. Example: poliovirus, adenovirus, hepatitis virus. Icosahedral capsid are stable and common in human pathogenic virus. Complex symmetry includes virus with more intricate structure. Example: pox virus, bacteriophages. Classification based on the nucleic acid of virus. The most commonly used system of virus classification was developed by Nobel Prize winning biologist David Baltimore in the early 1970s. The Baltimore classification schemes groups virus according to how the mRNA is produced during the reproductive cycle of the viruses into one of seven groups depending on a combination of their nucleic acid, either DNA or RNA. Viruses can contain double stranded DNA, single stranded DNA, double stranded RNA, single stranded RNA with a positive polarity, ssRNA with negative polarity, diploid 2-copy ssRNA and partial dsDNA genome. Positive polarity means that the genomic RNA can several directly as mRNA, and a negative polarity means that the required is complementary to the mRNA. Group 1. Viruses contain double-stranded DNA dsDNA as their genome. Their mRNA is produced by transcription in much the same way as with cellular DNA. Example: adenovirus, herpes virus, pox viruses. Group 2. Viruses have single-stranded DNA ssDNA as their genome. They converted their single-stranded genome into a dsDNA intermediate before transcription to mRNA can occur. Example: pox viruses. Then, Group 3. Viruses use dsRNA as their genome. The standards separate and one of them is used as a template for the generation of mRNA using the dependent RNA polymerase encoded by the viruses. Example: reoviruses. Then Group 4. Viruses have ssRNA as their genome with a positive polarity. Positive polarity means that the genomic RNA can serve directly as a mRNA. Intermediates. Intermediates of dsRNA called replicative intermediates are made in the processes of copying the genomic RNA. Multiple full-length RNA strands of negative polarity complementary to the positive stranded genomic RNA are formed from these intermediates, which may then serve as template for the production of RNA with positive polarity, including both full-length genomic RNA and shorter viral mRNAs. Example: coronaviridae, flavoviridae, astroviridae, picoronaviridae. Group 4. Viruses can contain ssRNA genome with a negative polarity. dsRNA intermediates are used to make copies of their genome and produce mRNA. Full-length positive RNA strands are made to serve as templates for the production of negative stranded genome. Example: orthomyxoviridae, paramyxoviridae, riboviruses. Group 6. Viruses have deployed 2 copies ssRNA genomes that must be converted using the enzyme reverse transcription to dsDNA. The dsDNA is the transported to the nucleus of the host cell and inserted into a host genome. The mRNA can be produced by transcription of the virus DNA that was integrated into the host genome. Example: retrovirus. In Group 7, viral have particle dsDNA genomes and make ssRNA intermediates that act as a mRNA but are also converted back into a dsDNA genomes by reverse transcription necessary for all genome replication. Example: hepadnaviruses. In this picture clearly explain the genetic material present in the virion. First, they have group 1 and group 2, group 3, group 4, group 5, group 6, and group 7. It's similar as mRNA, and mRNA produce the end product as protein. Protein is a coiled thread-like structure in nature. Group 1 is positive and negatively charged. Group 2 DNA contain positive charge. Group 3 RNA contain positive and negative charge. Group 4 RNA contain positive charge. Group 5 RNA contain negative charge. Group 6 RNA contain positive charge. Group 7 DNA contain positive and negative charge. During the group 6 and 7, in 6, reverse transcription is acquired. The DNA fragment is uncoiled and replaced. It also replicate in group 7. It also reverse transcription is acquired. The DNA is formed in reverse word. It will be the end product is protein. Host range and tropism. Host range, the variety of host or a virus can infect it. Tropism, specificity of a virus for certain tissues or cell types. Methods of classification, molecular techniques, PCR sequencing, serological methods, antibiotic detection, morphological techniques, electron microscopy. Application of viral classification. It could be easy to identify the classes of virus in certain groups. It would be able to classify its similar groups as possible as it will be clarity to study and useful to research purpose and another level of studies and research works. Thank you for your patient listening. Thank you one and all.